All right, my name is Hunter, and this is a review of the nervous system and muscle function. So, in the following questions, test your understanding of the various parts of the nervous system by asking yourself what would happen if some part was damaged. What would the system still be able to do? What would it be unable to do? Question 1. Some nerve gases and insect poisons work by destroying acetylcholine esterase. Acetylcholine esterase is normally present in acetylcholine synapses and acts to degrade acetylcholine. So, what is likely to happen to nervous? That's my dog in the background sighing. What is likely to happen to nervous transmission in insects exposed to this type of insect poison? So, in the central nervous system of vertebrates, acetylcholine can cause inhibition or excitation of the postsynaptic neuron. So when you have your, let's see here. So postsynaptic after the synapse, that space between neurons. Acetylcholine is responsible for inhibition or excitation. So which of these it does really depends on the type of postsynaptic receptor present. In neuromuscular junctions, such as voluntary muscle, acetylcholine is excitatory, and in heart muscle, the effect of acetylcholine is to reduce the rate and strength of heart muscle contractions. And so if acetylcholine esterase is destroyed, then any acetylcholine released into the synapse will stay there. And as a result, the postsynaptic neuron will be either continuously stimulated, as with the muscles, the muscle cell, or excuse me. So as a result, that postsynaptic neuron will either be continuously stimulated by acetylcholine to either depolarize if its receptor is excitatory, or hyperpolarize if its receptor is inhibitory. So for example, after an action potential is initiated in that muscle fiber, the muscle will contract continuously. By contrast, the heart rate will slow. So inability to control the contraction relaxation of muscles, such as the diaphragm, would obviously quickly result in death. So let's do a little bit of typing here. In central nervous system, acetylcholine can cause inhibition or excitation of the postsynaptic neuron. Acetylcholine esterase degrades acetylcholine, so any released into the let's see, into the synapse. So acetylcholine esterase degrades acetylcholine, so any released into the synapse would remain there, resulting, let's see. What is likely to happen to nervous transmission in insects exposed to this type of poison? All right, acetylcholine can cause inhibition or excitation of the postsynaptic neuron. Acetylcholine esterase degrades acetylcholine, so this type of poison would result in any acetylcholine released into the synapse remaining there, resulting in either constant excitation or suppression of an action potential. And this would result in death. Question 2, the pufferfish, fugu, contains the poison tetrodotoxin. Some shellfish produce a paralytic poison called saxitoxin. Both of these poisons block the sodium ion channels in neurons. What specific effects would these toxins have in neuron function. So remember in the last unit we were talking about sort of the salty banana of the structure of the neuron with potassium and potassium and sodium concentrations being different on either side of that membrane to help generate action potentials in the form of and generating nerve signals. So if the sodium channels were blocked, these axons would be unable to generate or propagate those action potentials. So if the sodium ion channels were blocked, axons would be unable to generate or propagate action potentials. Propagate. A type of spider, the funnel web spider, produces a toxin that blocks calcium channels. Can a neuron exposed to this toxin fire an action potential and explain? And the short answer here is yes. Neurons exposed to this toxin should still be able to generate action potentials since calcium isn't directly responsible for generating those action potentials. So yes. And part B, can a neuron transmit a signal across the synapse using neurotransmitters? And explain. So in chemical synapses, an influx of calcium ions is required to cause the synaptic vesicles to fuse with the membranes of the presynaptic neuron. So let's go ahead and write that because there's a lot to dig into here. So in chemical synapses, an influx of calcium is required to cause the synaptic vesicles to fuse with the membrane of the presynaptic neuron. If these calcium channels are blocked, no neurotransmitters are released into the synapse. If these calcium channels are blocked, no neurotransmitters into the synapse. Therefore, the signal cannot be transmitted across the synapse. All right, so a little throwback to last week's worksheet where we talked about the giant squid axons, which are part of why we know so much about how these function now. You isolate a section of giant squid axon and arrange an experiment so that you can change the solution bathing the axon. You insert an electrode into the axon and place another electrode outside the cell so that you can measure the potential across the cell membrane. With the axon bathed in normal extracellular fluid, you observed a resting potential of -70 millivolts and action potentials when stimulated that reached +55 millivolts. And you can see that information reflected here. Now let's say you change the solution bathing the neuron by increasing the potassium concentrations to 40 millimolar. What effect will this have on the neuron? For example, will it depolarize the membrane and make it easier to start an action potential? Or will it hyperpolarize the membrane and make it more resistant to starting an action potential? Or will it have no effect? And you can explain your answer here. All right, so we've got normal concentrations. This is our millimolar concentrations of sodium and potassium. And then we have our experimental concentrations inside and outside the neuron. So we have normal, experimental, sodium, potassium. And remember, again, we've got that salty banana. So sodium on the outside, potassium on the inside. You can see those concentrations reflected here. And then in our experimental concentrations, we're changing, it looks like just the potassium. Yes. Okay, so by increasing the potassium concentration, what effect would this have? So in answering these questions, keep these facts in mind. The axon's permeability to potassium ions in particular is much greater than to the axon's permeability is to potassium ions is much greater than to chlorine ions, for example, which is much greater than to sodium ions. So at rest, here. At rest, the concentrations of negative ions inside and outside the axon are approximately equal. Again, these are just things to keep in mind as we're answering our question here. The equilibrium potential for each ion is dependent on both its concentration gradient and on the electrical gradient. The equilibrium potential. So equilibrium potential, like let's go back to concepts like osmosis and things like that. These things will always different from osmosis, I apologize. The equilibrium potential is, these ions are always going to diffuse down their concentration gradient from areas of high concentration to areas of low concentration. But it also depends on this electrical gradient. So sodium and potassium ions, both positive, have different impacts on that electrical gradient, as would chlorine ions. So let's actually get into the core of this answer here. So potassium ions cannot be added without simultaneously adding a counterbalancing anion, for example, chlorine, as a result. The addition of potassium chloride adds both potassium and chlorine ions to the outside of the membrane. This addition disrupts both the potassium and the chlorine equilibrium potentials. And the axon is much more permeable to, well, okay, that's a lot of talking. Let's write this down. So potassium ions cannot be added without adding a counterbalancing anion, such as chlorine. The addition of potassium chloride adds both ions to the outside of the membrane, which disrupts their equilibrium potentials. The axon's much more permeable to potassium ions than to chlorine or sodium ions, and as a result, potassium moves much more quickly into the axon, moving down its electrical gradient as well, to reestablish its equilibrium potential. So therefore, the result will be to depolarize the axon and make it more likely to generate an action potential. Since the membrane is more permeable to potassium ions move more quickly into the axon. Down its electrical gradient. Since the membrane is more permeable to potassium ions move more quickly into the axon, back down its electrical gradient to reestablish equilibrium potential. Therefore, the result will be to depolarize the axon and make it more likely to generate an action potential. All right, so what would happen if instead of adding more potassium to the outside, you added more sodium to the fluid bathing the neuron? So instead of creating that positive charge inside, we're increasing that concentration of positive ions outside of the neuron. So as in part A, sodium ions cannot be added without adding a counterbalancing ion. For example, that chlorine. So the addition disrupts the sodium and chlorine equilibrium potentials, and as before, the axon is more permeable to chlorine than to sodium. So chlorine ions would move into and hyperpolarize the axon. All right. As before, the axon is more permeable to chlorine than to sodium. So ions would move into and hyperpolarize the axon. Now, it's also important to remember in both of these cases, A and B, that the baseline concentrations will eventually be restored by sodium-potassium pumps, but those require energy to move those ions against their concentration gradients. So this change, this depolarization and hyperpolarization, is going to be a short-term change just for the duration of that action potential. So how's the system organized? Might have to whip out the pen here and do some more color coding. Neurons are single cells composed of processes called dendrites. On our drawing here, we've got dendrites down here, these little bundles, that carry information to the cell body, and an axon that carries information away from the cell body. In simple terms, nerves are bundles of dendrites or axons, or mixed axons and dendrites. Ganglia are collections of cell bodies, and brains are collections of ganglia. So processing and integration of information occurs as a result of interactions within and between ganglia. So that's kind of our umbrella organization here. So let's look at a simple circuit in the nervous system. So responding to stimuli requires, at a minimum, reception of a stimulus, which triggers B, an action potential in a sensory neuron, which synapses with C, the central nervous system. So here we have our, so we have reception of a stimulus, triggers an action potential down the axon to a sensory neuron, which synapses with, we have our central nervous system here, CNS, spinal cord. The central nervous system then generates an action potential in a motor neuron going the other direction, which causes a response in a muscle or gland. So we're prompted here in the space to draw this minimum system, but it's already kind of there for us. So we have our stimulus, which arrives at these dendrites. A action potential travels up the axon to the cell body, which synapses with the central nervous system, which synapses with that motor neuron. Action potential travels down this axon to these dendrites at a muscle cell, presumably to trigger a response. So this is a very simple reflex circuit. It's a closed circuit. So in the blink of an eye response, or let's see, the blink of an eye response is one type of simple reflex response that occurs, for example, in response to water splashing up out of a sink or to a snowball aimed at your face. And when this occurs, you blink, and only afterwards do you realize that you blinked. What would you have to add to your diagram above to allow you to realize that you blinked? So you'd have to add an interneuron or another synapse to the sensory axon. Again, this was our sensory axon here on the top. That's labeled here. And that would have to lead to an interneuron which would carry the information to the brain to allow you to realize what happened. So there would have to be another synapse here traveling upstream into the brain for you to realize that this reflex had occurred. You'd need to add an interneuron or another synapse to the sensory axon that led to an interneuron which would carry the information to the brain so you could realize what happened. Why does it take you longer to realize why you blinked than it takes for the blink reaction to occur? So this simple reflex circuit, again, this is sensory to motor neuron, bypasses the brain pretty well. It's quote, cutting out the middleman, that interneuron, which allows reception and interpretation functions of the brain to occur basically instantaneously. This means that the protective response, the blink, can occur very, very quickly. Most such reflexes are designed to protect or ensure the livelihood of the organism. All right, so the simple reflex, sensor to motor neuron cuts out the middleman, the interneuron, cuts out the interneuron, which allows reception and interpretation functions of the brain to occur. So a little clarifying language here. Reception. So it's the interneuron that allows the reception and interpretation functions of the brain to occur. By cutting out the interneuron, it's just doing the reception response parts of that circuit. So this speeds up the protective response of the blink, which is designed to protect the organism. Or the reflex. We could just as easily say any other reflex. Okay. What composes the central nervous system, the CNS, and how does it differ in general form and function from the peripheral nervous system, the PNS? So the CNS is the integration and processing center. Let's go ahead and write that. CNS is the integration and processing center. Now, as such, it contains large collections of cell bodies and interneurons. Large collections of cell bodies and interneurons. By contrast, the PNS, the peripheral nervous system, contains sensory neurons. PNS contains sensory neurons which carry information from the central nervous system to the effectors. So the sensors to the CNS and the motor neurons. I may have skipped a line here, I'm sorry. The peripheral nervous system contains the sensory neurons which carry information from the sensors to the central nervous system, and motor neurons which carry information from the central nervous system to the effectors. So sensors to the central nervous system, that's afferent. Let's go ahead and write that. And motor neurons, which carry information from the CNS to the effectors, or efferent. So you'll see these words a lot, and that's kind of why I'm taking pains to include them here. Afferent generally means towards, and efferent generally means away from. So afferent towards the central nervous system, efferent away from the central nervous system. So effectors affect the desired outcome. All right, so the autonomic nervous system is subdivided into the sympathetic and parasympathetic systems. How do the general functions of each of these divisions interact to maintain homeostasis? So we're really integrating a lot of the different units that we've reviewed in this piece where we have an opportunity to. So I'm going to refer you to your textbooks here to figure 4315, which I do not have pulled up on my browser, but that will give you a lot of the context here to really help visualize these relationships. All right, so we have the two divisions of the autonomic nervous system. We have our sympathetic division and our parasympathetic division. The sympathetic division of the autonomic nervous system controls responses by the sympathetic nervous system are often called fight or flight responses. So fight or flight responses. These tend to upregulate conversion of glycogen to glucose for energy generation to cause secretions of epinephrine. Remember, we talked about those glands on the top of the nephron, the epi of the nephron. And to increase heart rate, blood pressure, and flow of blood to skeletal muscles and the brain. So this is very excitatory. It's amping up that response. So we're let's see, tend to upregulate of glycogen to glucose for energy generation. Secretion of, I'm just going to write epinephrine. Increase heart rate, blood pressure, and increase of blood flow to the skeletal muscles in the brain. So we're getting ready to fight, flight, or freeze. Really, there are additional responses in here that are better understood now, but this is pretty well established and helpful for really just understanding this division really well. So then we have our parasympathetic division. So the parasympathetic system acts antagonistically to the sympathetic nervous system, meaning it works against it for the most part. So antagonistic to the sympathetic division for the most part. When stimuli to the sympathetic system are removed, for example, stress or danger, then the parasympathetic system promotes the calming activity, so the rest and digest functions. You'll hear this called sometimes, rest and digest. When stimuli to the sympathetic system are removed, so these calming activities are going to look like decreasing heart rate and blood pressure, increased digestion and glycogen production, so increased digestion and glycogen production. One exception to this is in the control of the reproductive organs, where the sympathetic and parasympathetic systems complement each other, or they work in concert. One exception is the reproductive system. Both sympathetic and parasympathetic systems work in concert. All right, fibromyalgia is a condition that affects about 2% of the US population. Its primary symptoms are chronic pain, difficulty sleeping, and fatigue that is not relieved following sleep. Other reported symptoms include jaw clenching at night, dryness of mouth and eyes, an increased need to urinate, irritable bowels, muscle numbness and tingling, and headaches. Several researchers have proposed that fibromyalgia results from failed regulation of part of the autonomic nervous system. If this is true, failure of the regulation of which division of the autonomic nervous system could account for these symptoms? Explain your reasoning. So just given the symptoms alone, one could propose that the illness is caused by hyperactivity of the parasympathetic system. This would account for all of the symptoms, with possibly the exceptions of dry mouth and muscle problems and headaches. So symptoms suggest hyperactivity of the parasympathetic system, except maybe dry mouth, muscle problems, and headaches. To test their ideas, the researchers recorded the heart rates of both controls and individuals with fibromyalgia for 24-hour periods of normal activity. They observed that patients with fibromyalgia showed greatly elevated heart rates both while active and when asleep. Does the addition of this information change or support the hypothesis you developed in Part A? So given this information, the primary symptoms appear to be the results of the hyperactivity of the sympathetic system. So given this information, the primary symptoms appear to be the result of hyperactivity of the sympathetic system. Hyperactivity of the sympathetic system would elevate the heart rate and blood pressure and lead to difficulty sleeping and chronic fatigue. Prolonged exposure to high levels of adrenaline associated with hyperactivity of the sympathetic system could also lead to a reduced stress response. So let's write that down. Hyperactivity of the sympathetic system would elevate heart rate and blood pressure and lead to difficulty sleeping, chronic fatigue, prolonged exposure to high levels of adrenaline or epinephrine is associated with hyperactivity of the sympathetic system and also leads to a reduced stress response over time. So the researchers propose that this may be the cause of the irritable bowel and bladder as well as the limb numbness and dryness of the eyes and mouth. All right, so how does sarcomere structure affect muscle function? So how are sarcomeres arranged within muscle fibers and therefore within the muscle? So remember, a sarcomere is the unit of length, I guess, is the simplest terms to put this in. A unit of length in muscle fibers. So sarcomeres are arranged linearly in those myofibrils, myo being related to muscle. So sarcomeres are arranged linearly in myofibrils, muscle fibers, literally. Myofibrils are arranged linearly in muscle fibers, yeah. Muscle fibers are organized into muscles. So within a given muscle, the muscle fibers are all oriented in the same direction. So this is that arrangement of sarcomeres within muscle fibers. So Sarcomeres arranged linearly into myofibrils. Myofibrils arranged linearly in muscle fibers. Muscle fibers organized into muscles. And within those muscles, the muscle fibers are all oriented in the same direction. And as a helpful reference, you can check out figure 45.1 in your textbook. Question 2. Describe the sliding filament model of muscle action. For example, what interactions power the movement of filaments in association with each other? Include the troponin complex, calcium binding sites, tropomyosin, myosin binding site, and actin in your discussion. So I'm going to go ahead and shortcut this whole thing. There is a really fantastic Khan Academy video where they illustrate this really, really well. And I know that video was really helpful in helping me be able to visualize this entire process. You can also check out figure 45.2 through 45.6 in your textbook. So with that preamble, here we go. In the sarcomere, there are thick myosin filaments that lie between thin actin filaments. So in the sarcomere, thick myosin filaments lie between thin actin filaments. The actin filaments are attached to the Z lines of the sarcomere. The actin filaments of the sarcomere. The thick filaments contain myosin heads, which alternate between low and high energy states. In its low energy state, a myosin head is bound to an ATP molecule. The ATP gets hydrolyzed to generate the myosin head's high energy state. The actin filaments are composed of two strands. Two strands of actin intertwined to form a helix. Long strands of tropomyosin are wound around the actin helix. At rest, the tropomyosin strands block the myosin binding sites on the actin. Action potentials in the sarcoplasmic reticulum and T-tubules cause the sarcoplasmic reticulum to release calcium ions into the cytoplasm of the myofibrils. When that calcium binds to the troponin complexes, which remember are situated at intervals along the tropomyosin strands, they change configuration. The configuration change alters the position of the tropomyosin strands and exposes the myosin binding sites. The myosin sites hydrolyze ATP and pull the actin filaments toward the center of the sarcomere. That is towards the M line, and the muscle contracts. All right, so there's a lot going on there, but a helpful way to imagine this is kind of there's this grabbing on and letting go of the myosin head to the I'm going to leave it to the video illustrations here. This is a little bit too hard to illustrate with my current setup, since I can't do animations. But it is kind of crawling along, pulling those cables along to shorten the sarcomere towards that M line. All right, so how do your answers to question 1 and 2 help explain why at maximum contraction a muscle measures 70% of its extended or uncontracted length? So during contraction, the actin filaments are pulled toward the center of the sarcomere by the myosin heads on the thick filaments. So at maximum contraction, the sarcomere width is equal to the length of the thick filament. So at maximum contraction, the sarcomere width is equal to the length of the thick filaments. During contraction, the actin filaments are pulled toward the center of the sarcomere by myosin heads on the thick filaments. In the relaxed sarcomere, the length of the thick filaments is about 70% of the sarcomere width from the Z line to Z line. So let's in the relaxed sarcomere, the length of the thick filament is about 70%. From Z line to Z line. You can check this by measuring the widths of the relaxed sarcomere and the contracted sarcomere in figures 45.2. All right, review 45.6 and the associated text in Biological Science 7th edition, then answer these questions. Back to acetylcholine. Acetylcholine in an excitatory neurotransmitter is the neurotransmitter released into the neuromuscular junction. This release can trigger an action potential along the length of the muscle membrane. Describe the process involved. A motor neuron synapses with a muscle fiber at a neuromuscular junction. If an action potential is generated in the neuron, the motor end plate of the neuron releases acetylcholine, and this triggers an action potential in the muscle cell, which travels into the T-tubule system. And this causes the release of calcium ions from the sarcoplasmic reticulum, there we go, which then triggers contraction of the sarcomere. If acetylcholine always produces an excitatory response in the neuromuscular junction, how can we regulate which muscles, in the arm for example, are contracted and which are extended at any given time? For example, how can we bend the arm only partially? So this goes back to that arrangement of the muscle fibers themselves. Muscles are arranged into motor units. Muscles are arranged into motor units. The contraction of each motor unit is controlled by separate neurons. Separate neurons. The degree of response depends on how many motor units have been stimulated to contract. Severe calcium deficiency can lead to a reduction in bone mass. It can also have serious effects on the functioning of the nervous system and on the action of muscles. Explain what roles calcium plays in the activity of muscles. So as noted, it is the release of calcium from the sarcoplasmic reticulum that ultimately causes the muscles to contract. The release of calcium ions from the sarcoplasmic reticulum that ultimately causes the muscles to contract. In cases of severe calcium deficiency, muscles can become weak or incapable of strong contractions. All right, that was a lot of material. A lot of this is very visual, which unfortunately my current setup isn't optimized for. So I would really recommend Googling some videos on how these systems work, really taking a look at those images in your textbook. Thank you all for watching, and thank you for your flexibility this week. Life has been throwing a lot of curveballs lately, and I appreciate everybody's support and flexibility while we deal with that. I will be uploading this to YouTube momentarily, where you will be able to play this at whatever speed you like and review it at your convenience. Due dates should be extended. If they haven't, I will follow up on that and make sure everyone gets the credit that they are entitled to. Thank you all for your attention.